Hey guys, this is the second part of carbohydrates. Let's discuss about disaccharides and polysaccharides. Carbohydrates include simple sugar like monosaccharides or single sugar. This is an example on monosaccharide glucose. Glucose is a basic subunit of other larger carbohydrates molecules. Glucose is found in fruits, vegetables, and honey. Other examples of monosaccharides are galactose and fructose. Fructose is the sweetest of the sugars. It occurs naturally in fruits and honey, therefore we call it as fruit sugar. Fructose combines with glucose to form sucrose. Galactose combines with glucose to form lactose, or we call it as milk sugar. And now we are discussing about disaccharides. These are examples of disaccharides: sucrose, lactose, and maltose. Sucrose is known as table sugar, consists of glucose and fructose. Lactose is known as milk sugar, consists of glucose and galactose, while maltose or malt sugar consists of glucose and glucose. We need to understand how these monosaccharides join together to form disaccharides. The anomeric hydroxyl group and the hydroxyl group of a sugar compound can form a bond called a glycosidic bond by releasing water. This reaction is an example of the formation of glycosidic bond. A methanol reacts with the anomeric OH groups of glucose can form methyl glucoside or methyl glucopyranose. The joining and cleaving of sugar molecules are basically a condensation and hydrolysis reaction. In a condensation reaction, a carbon on one monosaccharide bonds to an oxygen on another monosaccharide, releasing one molecule of water. While in hydrolysis reaction, the disaccharides can break, and a water molecule supplies the hydrogen and hydroxyl group necessary for the existence of two complete monosaccharides. Okay, now let's discuss our first disaccharides, maltose. Maltose is a product of hydrolysis of starch. It's a disaccharide with glycosidic bonds between C1 and C4 of two glucoses. The glycosidic bonds is alpha. Cellobiose, a product of cellulose breakdown, is the otherwise equivalent of beta anomer. Oxygen on number C1 points up. The beta-1,4 glycosidic linkage is represented as zigzag, but one glucose is actually flipped over relative to the other. These are food containing maltose. They are bagels, sweet potatoes, and honey. They look delicious. The second disaccharide is lactose. About 5% of milk contains lactose, or about 50% in solid milk. Lactose does not occur elsewhere. Glucose-galactose is linked by beta-1,4 glycosidic bond. Galactose ring can open and close, therefore it's called reducing sugar. Lactase deficiency leads to lactose intolerance. These are food containing lactose. They are milk and the product containing milk, including cheese and yogurt. Next is sucrose. Sucrose is found in table sugar. It's obtained from sugarcane and sugar beets. Sucrose consists of glucose and fructose. Sucrose has an alpha-beta-1,2 glycosidic bonds, the bond between alpha-glucose and beta-fructose. Sucrose is not reducing sugar, since the anomeric carbon or carbon number 1 cannot open to give a free aldehyde to oxidize. These are the comparison of three disaccharides: maltose, lactose, and sucrose. Okay, now we are about to discuss about polysaccharides. Polysaccharides are large molecules of monosaccharides that are connected to each other through their anomeric carbons. There are two types of polysaccharides. One is storage polysaccharides, contain only alpha-glucose units. Three important from storage polysaccharides are amylose, amylopectin, and glycogen. Second is structural polysaccharides, contain only beta-glucose units. Two important ones are cellulose and chitin. Chitin contains a modified beta-glucose unit. Storage and structural polysaccharides are made up of glucose units, but they are structurally and functionally different because of their glycosidic bonds and differences in branching. These are our first storage polysaccharides. They are amylose and amylopectin, or they compose of starch. Starch is a mixture of amylose and amylopectin, and it is found in plant foods. Starch is abundant in rice and potatoes. Amylose makes up about 20% of plant starch, and it's made up of 250 to 4,000 T-glucose units bonded by alpha-1,4 in a continuous chain. Long chains of amylose tend to coil. Amylopectin makes up 80% of plant starch, and it's made up of T-glucose units connected by alpha-1,4 glycosidic bonds. The branch is connected through alpha-1,6 glycosidic bonds. These are the animation and the comparison of molecular structure of amylose and amylopectin. You can see that amylose has straight chain, while amylopectin has branched chain. About every 25 glucose units of amylopectin, a branch of glucose units are connected to the glucose by an alpha-1,6 glycosidic bonds. During fruit ripening, starch undergoes hydrolysis of the alpha-1,4 bonds to produce glucose and maltose, which are sweet. When we consume starch, our digestive system breaks it down into glucose unit or to use by our bodies. Another storage of polysaccharides is glycogen. Glycogen is a storage polysaccharide found only in animals. Glycogen is stored in the liver and muscles. Its structure is identical to amylopectin, except that alpha-1,6 branching occurs about every 12 glucose units. When glucose is needed, glycogen is hydrolyzed in the liver to form glucose. Alright, these are the comparison of molecular structure of amylose, amylopectin, and glycogen, so that you can visualize their differences. We then move on to structural polysaccharides. The first one here is cellulose. Cellulose contains glucose units bonded by a beta-1,4 glycosidic bond. This glycosidic bond configuration changes the three-dimensional shape of cellulose compared with that of amylose. The chain of glucose units is straight. This allows chains to align next to each other to form a strong rigid structure. The biological function of cellulose in plant is to provide shape and rigidity to the cell wall. In plants, cellulose is embedded within a matrix consisting of pectin and lignin. Pectin and lignin fill in any of the empty spaces found between rows of cellulose to help increase the strength of the cell wall. The cellulose chain align next to each other to form hydrogen bond intramolecular to give rigid and strong structure of the cell wall. Next is we talk about another storage polysaccharide. These are chitin. Chitin makes up the exoskeleton of insects and crustaceans and cell walls of some fungi. It is made up of N-acetylglucosamine containing beta-1,4 glycosidic bonds. It is structurally strong like cellulose. Chitin is also used as surgical thread that biodegrades as a wound heals. Chitin also serves as a protection from water in insects. Chitin is also used to waterproof paper and in cosmetics and lotions to retain moisture. Monomeric unit of chitin is modified beta-glucose that has N-acetyl D-glucosamine in C number 2. Chitin in the cell wall of bacteria gives a rigid and strong structure of bacteria cell wall. These are the comparison of structural polysaccharides: chitin, cellulose, and chitosan. Chitosan is the acetylated form of chitin. So now we have a clear picture of what carbohydrates are, from simple sugar like monosaccharides and disaccharides to huge polysaccharides with thousands of repeating units. This will come back later when we learn about metabolism. But for now, let's move on to another type of biomolecules, proteins, in our next video.